The difference is between the fungi and plants, green plants. Fungi are multinucleated, the cells are multinucleated, while the plant cell is uninucleated. Fungi multiply and reproduce through spores, while plants reproduce through seeds. The body of fungi are filamentous, while the body of plants are made of a variety of cells leading to tissue types and organs. Fungi are heterotrophic. They cannot produce their own food. They depend on already made food. While plants are autotrophic, they can manufacture their own food with their green pigmentation, the chlorophyll. The cell wall of fungi is made of chitin, while plant cell wall are made up of cellulose. So plant differentiations end in tissue types, but animals get to organs and systems. General mode of nutrition in fungi. Fungi lack chlorophyll, the green pigment that enables plants to make their own food. Consequently, fungi cannot synthesize their own food the way plants do. That's why plants are autotrophic while fungi are heterotrophic in nature. In order to feed, fungus releases. So fungi release digestive enzymes that break down food outside their bodies. The fungus then absorbs the dissolved food through the cell wall, its cell wall. Depending on that means they depend on outside sources for food. Fungi develops various living arrangement that enhances their opportunities for food absorption. Some fungi live as parasites, feeding on living plants, animals, and even other fungi. Certain fungi parasitize, injure plants and animals, causing millions of dollars of damage to farm animals, crops, and trees each year. For example, the fungus Ophiostoma ulmi, which causes Dutch elm disease, has killed tens of millions of elm trees around the world. Fungi that can obtain their food by breaking down dead organisms or substances that contain organic compounds such as starch and cellulose are called saprophytes, while those that decomposes organic materials, saprophytes that can also cause food spoilage and destroy wood products. There was a revolution during the revolution of in America, 1775 to 1783. British ships were destroyed by wood digesting saprophytes than by the enemy attack. Some saprophytes even grow in aviation fuels, where they break down the fuels, destroying their usefulness. So fungi also form highly specialized relationships with other organisms. For example, symbiosis. The roots of most plants develop mutual beneficial association with fungi to form what you call mycorrhizae. So mycorrhizae greatly increase the nutrition absorbing capacity of the plant roots. The fungus absorbs minerals from the soil and exchanges them for organic nutrients synthesized by the plant. Fungi also form leaves and bring them into their underground nests, where they feed them to certain fungi. These fungi primarily live in ant nests, and the ants eat nothing but the fungi. Some termites and wood-boring beetles use fungi to break down the cellulose in wood, making the wood easier for insects to digest. Plant parasites such as rust invade plant cells via specialized structures called heusteria that absorb nutrients from the cell. Fungi have characteristic and highly successful mode of nutrition. They need preformed organic compounds as a source of energy, as sources of energy. As sources of energy and as carbon skeleton for biosynthesis. The simplest of these compounds are monosaccharides, amino acids, fatty acids, etc., and are transported across the cell membrane. But more complex ones, which include many disaccharides, are degraded to monomers outside the cells by means of extracellular enzymes, which are released through the cell walls. Only the slime molds lack cell walls and are therefore able to engulf food and digest it intercellularly. Extracellular depolymerase enzymes of fungi and bacteria are used for many commercial purposes, which include the following. 1. Proteases used in producing washing powders and in food processing. Pectic enzymes in clarifying fruit juice. Lipase used in making cheese. Amylase used in hydrolysis of starch. Cellulase used in breaking down organic waste and in making some indigestion tablets. Little is known about the synthesis and release from fungi cells of these enzymes. These enzymes are known to be released when vesicles fuse with plasmalemma at the hyphal apex. Some then diffuse into the surrounding medium, while some remain within the wall to be released later. Many are induced by the presence of their substrate and the presence of more utilizable materials are known to repress their synthesis. The breakdown products released from complex polymers are available to all organisms because digestion in fungi occur in external environments. That means usually it is when the substrate, that is the material which the fungi grow in, that stimulates the enzyme to be released. in most fungi, whilst some are not stimulated by such a environment or in such environments. Some are actually repressed by such environments. Some fungi seem to be specifically adapted to grow within the zones of substrate breakdown created by other fungi. That means when other fungi release their enzymes, then it will attract some other fungi to grow on the enzymes released by another fungi. So, and they will absorb some of the nutrients available in the environment. So for extracellular digestion to take place, you must have moisture. Moisture must be available. Water films are required for the diffusion of nutrients and enzymes. So fungi can only grow in relatively moist environments. So the wall of actively growing fungi must be permeable to water. So they are particularly sensitive to desiccation. So when you are growing fungi, especially the macrofungi, and there is not enough moisture in the growing environment for it to use, then the growth will not be possible. Okay, if there is no question, I'll continue. The extracellular enzymes are released into an uncontrolled environment. The plant pathogen, for example, Sclerotium rowifi, secretes large amount of oxalic acid, which lowers the pH of the plant tissues where they infect, at which the fungi pectic enzyme works best. The enzyme degrades the middle lamella between plant cell walls, infecting the plant. Sepula lacriaman, which is the causal organism of dry rot of timber, degrades cellulose to glucose to form carbon dioxide and water, causing the wood to release metabolic water. The fungi utilize the water to colonize the dry timber, making the pathogen difficult to eradicate, because as long as there's presence of that water, the fungi will continue to grow and destroy the timber. Fungi degrade complex insoluble polymers to create zones of substrate exhaustion. Example of zones of enzymatic erosion around their cells, causing them to continually grow into fresh zones of nutrients. Cytoplasm in the old zones become redundant. This may be the main reason for apical growth. The apex grows wall into fresh zones much faster than it can duplicate its own cytoplasm, because cytoplasm moves into the apex from the zones of substrate exhaustion. Carbon and energy sources. There is extremely wide range of compounds that can serve as sole carbon and energy sources for fungi. The only common and naturally occurring compound that cannot be utilized by fungi is methane. A fungi can utilize hydrocarbon and alcohol like methanol and phenol, and very few can utilize more complex polymers like lignin and keratin. Consequently, such substrates only support a small and characteristic fungal flora. Carbohydrates of plant origin often form the most abundant energy source of fungi nature. Almost all fungi utilize maltose, sucrose, or starch wall Some can grow in the absence of sugar, or example, Leponitis lacteus. Leponitis lacteus is an oomycetes known as sewage fungus. Thus, fungus cannot utilize glucose, but that is, this fungus cannot utilize glucose, but relies on fatty acids, organic acids, and glycerol as its main source of energy. Fungus also utilizes other hexox or pectose sugars and derivatives like the uronic acid and sugar alcohol. Although fungi are limited in their ability to transport these materials across cell membrane, other substrates are fats, proteins, and many structural polysaccharides such as cellulose and hemicellulose, as long as the fungus can produce the necessary enzymes. Cellulose is particularly important in this respect, as it is the most component of plant cell walls. But even relatively simple molecules require the action of at least 3 enzymes before it is hydrolyzed to glucose and can enter the cell. The other aspect of nutrition in fungi is hydrocarbon and man-made materials. That means fungi utilize carbon and the energy sources, other energy sources, then they utilize hydrocarbon and man-made materials. Fungi can also utilize hydrocarbon and man-made materials, so fungi are cause nuisance in manufacturing factories, industries, where they grow on machines, oils, and other lubricants. If used as emulsion and cause clogging of filters, increase in viscosity of the lubricant, and corrosion. Fungi also grow on kerosene, another component of aircraft fuels. Examples of such fungi are Amophyteca racinae and Psilomyces varroitii. Both are Deuteromycetes. Deuteromycetes are the fungi imperfecti. Fungi also grow on paint, films of paint, causing discoloration. A very good example is Cladosporum species, which are seen on emulsion paints, films in bathrooms and kitchens, and even herbarums, where their appearance is usually described by their collective common name, sooty mold. The fungi derive energy from the soluble cellulose derivatives like carboxymethyl cellulose, which are incorporated into emulsion paints as thickeners and to stabilize the emulsion. There is this attention drawn to plastic, pesticides, and detergents as substrates for fungi because they are major pollutants. But the major component of plastic cannot be utilized by fungi, basic microorganisms, but some macrofungi has been proven to be useful in degradation of plastic, pesticides, and detergents.